Today we're going to talk about an overview of the glycolysis and gluconeogenesis pathways. So first let's just do a little bit of history. Who figured it out? Edward Buchner is on the top, who discovered the cell-free fermentation, and Otto Meyerhoff is on the bottom. He was also involved in completing the puzzle of glycolysis. So the pathway of glycolysis, as it is known today, took about 100 years to discover. So the combined results of smaller experiments were required in order to understand the pathway as a whole. So the first step is understanding glycolysis, and that began in the 19th century with the wine industry. So for economic reasons, the French wine industry sought to investigate why wine sometimes is distasteful, and instead of fermenting into alcohol. So the French scientist Louis Pasteur researched this issue in the 1850s, and his experiments showed what began to be a long road in actually eluding the pathway of glycolysis. And his experiments showed that fermentation occurs by the action of living microorganisms, and we all know, we've all heard of yeast. So the yeast glucose consumption decreased under aerobic conditions of fermentation in comparison to anaerobic conditions. And Edward, the one on the top of our slide, discovered the cell-free fermentation. So while Pasteur's experiments were groundbreaking, insight into the component steps of glycolysis were provided by the non-cellular fermentation experiments during the 1890s. And he also demonstrated that the conversion of glucose to ethanol was possible using a non-living extract of yeast. And this is really just due to the action of the enzymes that are in the yeast. So the experiment not only revolutionized biochemistry, but it also allowed later scientists to analyze the pathway in a more controlled lab setting. So in a series of experiments from 1905 to 1911, some other scientists, Arthur Harden and William Young, discovered more pieces of glycolysis. They actually discovered that the regulatory effects of ATP on glucose consumption during fermentation. So they shed light on the role of one compound as a glycolysis intermediate, fructose. And the discovery of fructose, which is a 1,6-biphosphate, was accomplished by measuring carbon dioxide levels when yeast juice was incubated with glucose. So carbon dioxide production increased rapidly, and then it slowed down. So Hayden and Young noted this process would start in an inorganic phosphate, and it was added to the mixture. So Harden and Young concluded that this process produced organic phosphate esters, and further experiments followed those. So Arthur Harden and William Young, along with Nick Shepard, determined in a second experiment that a heat-sensitive, high-molecule subcellular fraction, the enzymes, and a heat-intensive, low molecular weight cytoplasm fraction, which is ADP, ATP, and NAD, and other cofactors are required together for fermentation to proceed. So the experiment began by observing that purified yeast juice could not ferment or even create a sugar phosphate. So this mixture was rescued with the addition of undialyzed yeast extract that had been boiled. So the boiling the yeast extract got them all the proteins as it denatured them. And the ability of this boiled extract juice completed fermentation. It suggests that the cofactors were non-protein in character. Then there was Otto Meyerhoff, one of the main scientists involved, and this was in the 1920s, and he was able to link together some of the little individual pieces of glycolysis discovered by the others. And his team, they were able to distract glycolysis enzymes from muscle tissue, and they combined them artificially to create a pathway from glycogen to lactic acid. So in one of the papers that they did, they investigated the reaction that splits fructose, the 1,6-diphosphate, into two triose phosphates. And previous work had thought that the split occurred at the 1,3 plus an oxidizing enzyme and coenzyme. So they found that the equilibrium constant for this isomerase and alderoses, the reaction was affected by inorganic phosphates or any cosamase or oxidizing enzymes. And they further removed the disphosphoglyceride as an intermediate in glycolysis. So with all these pieces that were available by the 30s, Gustav Emden promised a detailed step-by-step outline of the pathway that we know today as glycolysis. So the biggest difficulties in determining the pathway were due to the very short lifetime and low, steady concentration of intermediates of the fast glycolytic reactions. So by the 40s, other biochemists had completed the puzzle of glycolysis, and the understanding of the isolated pathway has been expanded in the subsequent decades to include details of its regulation and its integration with other metabolic pathways. I think it's always great to look at a little bit of the history of how we got somewhere. So, what is glycolysis? So let's just start with that. It is derived from glycose, which is just an older term for glucose, and lysis degradation, which basically means that during the lysis, the protection of the cell membrane is destroyed. So glycolysis is a metabolic pathway that converts glucose into pyruvate and a hydrogen ion. So the free energy is released in this process, and it's used to form the high energy molecules ATP, which is adenosine triphosphate, and NADH. So glycolysis is a sequence of 10 enzyme catalyzed reactions. So most monosaccharides, such as fructose and galactose, can be converted into one of these intermediates. So an intermediate may also be directly useful rather than just utilized in the steps in the overall reaction. So for example, the intermediate DHAP is a source of the glycerol that combines with fatty acids to form fat. And glycolysis is an oxygen-independent metabolic pathway. So the wide occurrence of glycolysis indicates that it is an ancient metabolic pathway. So indeed, the reactions do constitute glycolysis and its parallel pathway. The pentose phosphate pathway occur metal catalyzed under the oxygen-free conditions of Archean oceans. So this is also in the absence of enzymes. So in most organisms, glycolysis occurs in the cytosol. So the glycolysis pathway can be separated into two phases. The preparatory, or it's sometimes called the investment phase, and this is where the ATP is consumed, or what is sometimes called the payoff phase, where the ATP is produced. Okay, so let's just go over the 10 steps of glycolysis first. So glycolysis is the systematic breakdown of glucose and other sugars to power the process of cellular respiration. It's a universal biochemical reaction that occurs in every living unicellular and multicellular organism, which respires aerobically and anaerobically, which basically means with oxygen and without. So there are many metabolic pathways through which this process occurs, and the glycolysis steps that are presented here refer to a particular process called the Embden-Meyerhof-Parnas pathway, which is named after the scientists, of course. So this process is a small part of cellular respiration and overall body metabolism that's directed towards creating ATP, which is the energy currency of the body. So glycolysis literally means glucose breakdown or decomposition. So one glucose molecule is completely broken down to yield 2 molecules of pyruvic acid, 2 molecules of ATP, and 2 NADH radicals, and carrying electrons. So it took years of painstaking research, we said 100 years, in biochemistry to reveal these steps. So I just want you to appreciate how long it took to actually figure this out. And I'm sure we will figure out even more in the future. But the whole process involves 10 steps with a product that's forming at every stage, and every one of them is regulated by a separate enzyme in each phase. So the production of various compounds at every step offers different entity points into the process. So that means that the process may directly start at an intermediate stage if the compound that is a reactant at that stage is directly made available. So in step 1, the phosphorylation of glucose, which is adding of a phosphate group, is what happens. The reaction is made possible by the enzyme hexokinase, which separates one phosphate group out of ATP. So in the process, one ATP molecule, which is the energy currency of the body, is used up and gets transformed to ADP. So due to the separation of one phosphate group, this is what happens. And the entire reaction can be summarized as glucose plus ATP plus hexokinase will actually yield glucose 6-phosphate and ADP. All right, next let's move to step 2. This is the production of fructose 6-phosphate, and it is made possible by the action of the enzyme phosphogluconeogenesis. It acts on the product of the earlier step, glucose 6-phosphate, and it transforms it into a fructose 6-phosphate, which is its isomer. And isomers are just different molecules with the same molecular formula, but a different arrangement of atoms. So the reaction is summarized as follows. Glucose 6-phosphate plus the enzyme yields the fructose 6-phosphate. So let's move to step 3. The production of fructose 1,6-diphosphate. In the next step, the isomer fructose 6-phosphate is it's converted to fructose 1,6-diphosphate by the addition of another phosphate group. So this conversion is made possible by the enzyme phosphofructokinase, which utilizes one more ATP molecule in the process. Okay, the 4th step, the enzyme aldolase, brings about the splitting of fructose 1,6-diphosphate into 2 different sugar molecules that are both isomers of each other. And the 2 sugars formed are glyceraldehyde phosphate and dihydroxyacetone phosphate. Step 5 is the interconversion of 2 sugars. I-hydroxyacetone phosphate is a short-lived molecule As soon as it's created, it's converted into glyceraldehyde phosphate by the enzyme called triose phosphate. So in total, the fourth and the fifth steps of glycolysis yield 2 molecules of the glyceraldehyde phosphate. So in step 6, it involves 2 important first reactions. First is the formation of NADH from NAD plus, and by the use of the enzyme triose phosphate. And the second is the creation of the 1,3-diphosphoglyceric acid from the 2 glyceraldehyde phosphate molecules produced in the earlier steps. Step 7 is the production of ATP and 3-phosphoglyceric acids. So the 7th step involves 2 ATP molecules along with 2 molecules of the 3-phosphoglyceric acid from the reaction of phosphoglycerokinase on 2 product molecules of 1,3-diphosphoglycerol acid, which was yielded in the previous step. And 8, the relocation of the phosphorus atom. Step 8 is a very rearrangeable reaction, which involves the relocation of phosphorus atom and 3-phosphoglyceric acid, the 3rd carbon in the chain, to the 2nd carbon and creates 2-phosphoglyceric acid. So the entire reaction is summarized as follows. 2 molecules of the 3 acid plus the enzyme, yields 2 molecules of the phosphoglyceric acid. And then in step number 9, this is where the removal of the water takes place. The enzyme enolase comes into play and removes the water molecule from the 2-phosphoglyceric acid to form another acid called phosphophenol pyruvic acid, PEP. This reaction converts both of these molecules to 2-phosphoglyceric acid that form in the previous step. Okay, step 10, the creation of pyruvic acid. This step involves the creation of 2 ATP molecules along with 2 molecules of pyruvic acid from the action of enzyme pyruvate kinase on the 2 molecules of phosphophenol pyruvic acid produced in the previous step. So this is made possible by the transfer of a phosphorus atom from phosphophenol pyruvic acid to ADP molecules of the phosphophenolpyruvate acid. So as you can see, all the steps mostly involve the manipulation of a phosphate group and then the phosphate atom, which is made possible by various enzymes in the cytoplasm. The enzymes are like catalysts, which make a reaction possible and then disintegrate. So in the end, the whole process involves the breakdown of 1 glucose molecule that will yield 2 molecules of NADH, 2 molecules of ATP, 2 molecules of water, and 2 molecules of pyruvic acid. So the product of glycolysis are further used in the citric acid, or what we call the Krebs cycle, which is a part of cellular respiration. Okay, so now we're going to talk about intermediates or other pathways. So one of the articles that I was reading concentrated on the catabolic role of glycolysis with regard to converting potential chemical energy to usable chemical energy during the oxidation of glucose to pyruvate. So Many of the different metabolites in the glycolytic pathway are also used in anabolic pathways, and therefore the flux through the pathway is critical to maintain the supply of carbon skeletons for biosynthesis. So these are some of the pathways that are strongly reliant on glycolysis as a source, and there are many more, but there is, for example, like the pentose phosphate pathway, which begins with the dehydrogenation of glucose 6-phosphate, the first intermediate to be produced by glycolysis, and it produces various pentose sugars and NADPH for the synthesis of fatty acids and cholesterol. Glycogen synthesis also starts with the glucose 6-phosphate at the beginning of the glycolytic pathway. So glycerol for the formation of triglycerides and phospholipids is produced in the glycolytic intermediate glyceraldehyde 3-phosphate. And various post-glycolic pathways, fatty acid synthesis, cholesterol synthesis, the citric acid cycle, when it which in turn will lead to amino acid synthesis, nucleotide synthesis, tetrapyrrole synthesis, and although glucogenesis and glycolysis share many intermediates, the one is not functionally a branch or a tributary of the other part. There are 2 regulatory steps in both pathways, which, when active in one pathway, can automatically inactivate the other one. So the 2 processes cannot be simultaneously active. If both sets were highly active at the same time, the net would be the hydrolysis of 2 ATP and 2 GTP per reaction cycle. And NAD is the oxidizing agent in glycolysis, as it is in most other energy yielding metabolic reactions. And the NADH is produced primarily used ultimately to transfer electrons to oxygen to produce water, or when oxygen is not available to produce compounds such as lactate or ethanol. So NADH is rarely used for synthetic processes, the notable exception being gluconeogenesis, which we're going to talk more about in a minute. So being a fatty acid and cholesterol synthesis, the reducing agent is NADPH. And the difference exemplifies a general principle that NADPH is consumed during biosynthetic reactions, where NADH is generated in energy yielding reactions. So the source of the NADPH is twofold. When malate is oxidatively decarbolized by NAD-linked malic enzyme pyruvate. So CO2 and NADPH are formed, and NADPH is also formed by the pentose phosphate pathway, which converts glucose into ribose, which can be used in the synthesis of nucleotides and nucleic acids, or it can be catabolized to pyruvate. Okay, let's look at glycolysis and disease. So we're going to see diabetes, genetic diseases, and cancer. So cellular uptake of glucose occurs in response to insulin signals, and glucose is subsequently broken down through glycolysis, which lowers the blood sugar levels. However, low insulin levels seen in diabetes result in hyperglycemia, whereas glucose levels in the blood rise and glucose is not properly taken up by the cells. Hepatocytes further contribute to this hyperglycemia through gluconeogenesis, and glycolysis and hepatocytes controls hepatic glucose production, and when glucose is overproduced by the liver without having a means of being broken down in the body, then hyperglycemia will result. On some of the genetic diseases, the glycolytic mutations are generally rare due to the importance of the metabolic pathway. So that means that the majority of occurring mutations result in an inability for the cell to respire, and therefore that causes the death of a cell at an early stage. So some mutations are seen, with one notable exception being the pyruvate kinase deficiency, which leads to chronic hemolytic anemia. And then in cancer, malignant tumors perform glycolysis at a rate that is 10 times faster than non-cancerous tissue, and therein lies the problem. So during their genesis, limited capillary support often results in a decreased oxygen supply within the tumor cells. And the cells rely on anaerobic metabolic processes such as glycolysis or ATP. And some tumor cells overexpress specific glycolytic enzymes, which result in higher rates of glycolysis. And often these enzymes are isoenzymes of traditional glycolysis enzymes. And these will vary in their susceptibility to the traditional feedback inhibition. And the increase in the glycolytic activity counteracts the effects of hypoxia by generating enough ATP from this anaerobic pathway. This was first described back in the 1930s, and the Warburg hypothesis claimed that cancer is primarily caused by dysfunctionality in mitochondrial metabolism rather than because of the uncontrolled growth of cells. A number of theories have been advanced to explain the Warburg effect. One such theory suggests that increased glycolysis is a normal protective process of the body, and that malignant change could be primarily caused by energy metabolism. So the high glycolysis rate has important medical applications as high aerobic glycolysis by malignant tumors is utilized. Clinically, they use this to diagnose and monitor treatment responses of cancers by imaging the uptake of the 2,18F2 deoxyglucose, which is basically just a radioactive modified hexakinase substrate with position emissions tomography. So there's ongoing research, like there always is, to affect the mitochondrial metabolism and treat cancer by reducing the glycolysis, thus starving out the cancer cells in various new ways, including the ketogenic diet. So, what is gluconeogenesis? So, it is a metabolic pathway that results in the generation of glucose from certain non-carbohydrate carbon substances. So, from the breakdown of proteins, these substrates include glucogenic amino acids, although not ketogenic amino acids. And from the breakdown of lipids, such as triglycerides, they include glycerol, odd-chain fatty acids, not the even-chained, and from other steps in metabolism, they include pyruvate, and lactate. So gluconeogenesis is one of several main mechanisms used by humans and many other animals. We use it to maintain our blood glucose levels, being able to avoid the very low levels like hypoglycemia. So, and other means, it will include the degradation of glycogen or glycogenolysis and fatty acid catabolism. So, gluconeogenesis is a process that's also present in plants, animals, fungi, bacteria, and other microorganisms. In vertebrates, it takes place mainly in your liver, to a lesser extent in the cortex of the kidneys. In ruminants, it tends to be a continuous process, and in many other animals, the process occurs during periods of fasting, starvation, low carbohydrate diets, or intense exercise. So the process is coupled. It's highly enderogonic until it's coupled to the hydrolysis of ATP or GTP, which makes the process exerogenic. So for example, the pathway leading from pyruvate to glucose 6-phosphate requires 4 molecules of ATP and 2 molecules of GTP to proceed spontaneously. This is what we hear often, and you hear this a lot with people dieting today or people with diabetes. You hear it being associated with ketosis. It's also a target of therapy for type 2 diabetes, such as the anti-diabetic drug metformin, which is a fairly common tablet or prescription that people take, which inhibits glucose formation and stimulates glucose uptake by the cells. But now in ruminants, the dietary carbohydrates tend to be metabolized by the rumen organisms. So in them, it occurs regardless of fasting or regardless of the low carbohydrate diet, etc. And that's a really nice diagram on the slide there for you to look at. So where is gluconeogenesis? Well, in mammals, it's been believed to be restricted to the liver, the kidneys, the intestines, and the muscles. But there is recent evidence that it has occurred in the astrocytes of the brain. And these organs are somewhat of a different precursor. The liver uses lactoglycerol and gluconeogenetic amino acids, especially alanine, while the kidney uses lactate, glutamine, and glycerol. So lactate from the cycle is quantitatively the largest source of substrate by gluconeogenesis, especially for the kidney. The liver uses both to produce glucose, whereas the kidney only uses the gluconeogenesis. So after a meal, the liver shifts to glycogen synthesis, where the kidney increases the glucogenesis. So gluconeogenesis is a pathway consisting of a series of 11 enzyme catalyzed reactions. And the pathway will begin in either liver or the kidney, in the mitochondria, or the cytoplasm of those cells. This is dependent on the substrate being used. And many of the reactions are the reverse of the steps in glycolysis. So glucogenesis begins in the mitochondria with the formation of oxaloacetate by the carbonylation of pyruvate, and the reaction also requires 1 molecule of ATP and is catalyzed by pyruvate carboxylase, and the enzyme is stimulated in high levels of acetyl coenzyme A, and it's inhibited by high levels of ADP and glucose. So this will just kind of give you an overview of how that is reversed. So glucose 6-phosphate is formed from fructose 6-phosphate by phosphoglucosaminase, which is the reverse step in 2 here of glycolysis, and glucose 6-phosphate can be used in other metabolic pathways to free glucose. So when free glucose can easily diffuse in and out of a cell, the phosphorylated form is locked in the cell, a mechanism by which intercellular glucose levels are controlled by cells. And the final gluconeogenesis, the formation of glucose, occurs in the lumen of the endoplasmic reticulum, where glucose 6-phosphate is hydrolyzed by glucose 6-phosphate to produce glucose and release an inorganic phosphate. So, like some of the steps prior, the reversal of glycolysis, in which hexokinase catalyzes the conversion of glucose and ADP into G6P and ADP. Glucose is shuttled into the cytoplasm by glucose transporters that are located in the endoplasmic reticulum's membrane. So while most steps in glucogenesis are the reverse of those found in glycolysis, 3 regulated and strongly androgenic reactions are replaced with more kinetically favorable reactions. Enzymes are typically regulated by similar molecules, but with opposite results. So for example, acetyl coenzyme A and citric activate the glucogenesis enzymes, while at the same time they inhibit the glycolytic enzyme pyruvate kinase. So this is what they call a system of reciprocal control, allowing glycolysis and glucogenesis to inhibit each other and prevent a futile cycle of synthesis. Glucose to only break it down. The major of the enzymes are responsible for gluconeogenesis are in the cytosol, with the exception being the mitochondrial pyruvate carboxylase, and in animals, the phosphoenol pyruvate carboxynase. And the latter exists on an isosome located in the mitochondria and the cytosol. So the rate of glucogenesis is ultimately controlled by the action of a key enzyme, which is regulated through signal transduction by CAMP and its phosphorylation. So global control of gluconeogenesis is released when the blood glucose is low. So it triggers phosphorylation of enzymes and regulatory proteins by the protein kinase A, which results in the inhibition of glycolysis and stimulation of gluconeogenesis. So insulin, which is the example I put on the slide here, type 2 diabetes, let me back up. The inhibition of glycolysis and the stimulation of glucogenesis. So insulin counteracts glucagon by inhibiting the gluconeogenesis. So type 2 diabetes is marked by excess glucagon and insulin resistance from the body. So insulin can no longer inhibit the gene of the enzymes such as PEPCK, which leads to increased levels of hyperglycemia in the body. And the anti-diabetic drug metformin that we talked about reduces blood glucose primarily through the inhibition of glucogenesis, overcoming the failure of insulin to inhibit glucogenesis due to insulin. So studies have shown that the absence of hepatic glucose production really has no major effect on the control of fasting plasma glucose concentrations, but the compensatory induction of glucogenesis occurs in the kidneys and the intestine, and is driven by glucagon and glucocorticoids and acidosis. And here are my references. And if any of my biology students at Pecola High School happen to have actually made it all the way through this video, give me the word zebra and I'll give you some bonus points. Thank you.